Hey plant taxers, imagine we're all together in Founders Hall, room 118. We're going to do lecture 16 together. We're going to be talking about variation, and I'm going to try to get through this within about 20 minutes or so. So first of all, I want to differentiate these two terms, diversity and variation. Diversity is when we're talking about differences at or above the species level. So we're looking at the species diversity, the richness of an area, or the diversity of the redwood forest. That's talking about the number of species. Variation is within a species. So in other words, infra-specific differences within just a single species. That's what we're going to be talking about today, variation. So variation, what does it mean to you? What causes it? Where do you find it? These are all topics we're going to talk about, kind of starting with the where do you find it part. Well, you find it at various levels. You find it among parts within a single individual, right? So if you look at different, say, leaflets along a leaf, they're going to be different from one another sometimes. We also see differences among individuals within a population. So you've got this field of flowers, and some of the flowers are bigger than others, something like that. Also, we're going to see variation among populations within a species. Okay, we're going to talk about all those things, starting with variation among parts within a single individual. Here we have a picture of the buttercup. This is in the genus Ranunculus. We'll actually be picking this up in lab very soon. Notice that we've got a few different kind of looks of leaves. This one at the bottom right down here, much bigger than, say, these really narrow-leaved ones way up here, and then this one's even smaller. So this is all along a single individual, but we're seeing kind of different shapes, different sizes. Well, this is what they look like kind of in silhouette version. This is the very first leaf to emerge at the base there. This is the second highest up, third, fourth, fifth, six, and then the very last one doesn't even have any lobes. Okay, cool. Somewhere else that we can see variation among parts is on another genus that we're about to pick up pretty soon. This is a eucalyptus tree, and here you can tell we've got a bunch of leaves. They're sort of curved, linear. Well, here's the truth about eucalyptus trees. All of those curved, linear leaves, they're only found on the older, kind of more matured trees. Younger individuals have leaves that look like this stem. So this is just a stem right here, and then each one of these are leaves, meaning we've got opposite leaves. These are the younger ones. These are the juveniles. Over here, and this can be literally on the exact same individual, but on a different part of the tree. This is kind of the older stuff. This is the older plant material. And here's a stem. Notice we've got, alternate leaf arrangement. They're not opposite. So we have different leaf arrangement. There's an obvious difference in the leaf shapes, the leaf colors even. The juvenile leaves, notice they've got that waxy coat. Well, you can kind of tell they're bluish. They've got that waxy, powdery coating. They're very glaucous leaves. Crazy. All right. Next slide is another picture of two stems you could find on just a single individual. This is a picture of canyon live oak, Quercus chrysolepis, and sometimes on the same plant you can have these leaves that have kind of entire margins, nice smooth edges, and then right next to it there's a different stem that's got these really coarsely dentate leaf margins, and these, we can't feel them from this picture right now, but I'll tell you, they are extremely rigid and sharp and needle-like. You do not want to walk through a bunch of canyon live oak that have that leaf margin. But the point of this picture, you can have variation among parts within a single individual. Here's another one. This is a picture of something you know. This is the Redwood Sequoia sempervirens. And what these numbers up here mean is these are the height that each sample was taken at. So this largest one, someone took a branchlet at only 2 meters off of the ground. This one that's much smaller was taken 55 meters off of the ground. These ones were taken at the very tops of the trees, 105 meters tall. And notice the different leaf morphology. This is all on one individual tree. The leaves at the base of the tree are much broader. At the top, the leaves are narrower. They're they're pointing in a different direction. They're kind of all shaped. Differences within an individual. We're also going to see differences among individuals within a single population. So now we're talking kind of broader variation. Here, this graph kind of represents the number of individuals, so the frequency. And let's say for this variable leaf length amongst, say, this population of oaks or something, we have most of the leaves that are in this population are kind of the middle of the run length. They're kind of medium length. Very few individuals have leaves that are very long. Again, very few individuals have leaves that are very short. And we kind of have this natural bell curve where most of the individuals in a population have the average, say, leaf length or a petal length, whatever the structure is that we're looking at. So here is a picture of a member of the Brassicaceae in a genus called Draba, and what we've got here are different individuals. Notice this one here, it's got a bunch of basal leaves. It's got these 2 or actually even 3 stems coming up, lots and lots of leaves and flowers and fruits, while these individuals back here, and these are entire individuals that are not connected to one another. They're all from different seeds. They're all different seedlings. This only has 1 stalk. And it's much, much, much smaller in general. The leaves, the peduncle, the number of flowers, everything's much smaller in those individuals. So this is all one population of, say, plants growing at the dunes or something like that. Okay. So as a botanist, you must expect variation amongst individuals. You must measure more than one individual, more than one leaf. Never be satisfied. Don't be content measuring just one leaf. Measure as many as possible if you've got them available. Because something else you might see is something like this. Here we have variation within an individual and between individuals. So these, the top row here, the top row of leaves are from one plant. And notice within the single plant, we've got some leaves that are really big, some leaves that are really small. Same thing with plant number 2. Again, an unconnected individual of the same species. We've got some leaves that are much smaller, some leaves that are much bigger. But between these 2 individuals, we have drastically different leaf shapes. Plant number 2 has these really narrow leaves, more kind of lanceolate, while plant number 1 has these much broader, more ovate leaves. Okay, cool. So variation within an individual and between individuals. We also see variation among individuals within a population on less of a gradient. Sometimes we see just a drastic individual difference and no kind of intermediate. This is a picture of Vaccinium parvifolium, the huckleberry. And what we see here is instead of, like I said, that gradient, that bell curve, we just see kind of 2 different variants, binary, red or white here. And this might be considered albino. Here's another example of a plant that we see that kind of every once in a while just has these albino flowers. This is called the sweet pea. It's a weedy invasive plant that you see here in Arcata and lots of other places in the world. We have to know about this variation within a species. We've got to know. Sometimes, rarely, albinos occur. It's always good to know these things as a botanist. The last kind of level that I wanted to talk about where we see variation is among populations within a species. So here's a population. Let's say some oak species. We've got some oak trees that are kind of the average height. We have other oak trees that are really, really tall and some that are kind of much more smaller. Well, there's different populations of this oak species around California. And in some places, let's say, oh, the maximum height of a tree is much, much taller than the maximum height in a different place in California. And here the average is also kind of much, much taller than the average in, say, a different population within this species. Okay, why are we talking so much about variation? Well, variation is the raw material that natural selection can work on. It's the basis of evolutionary change. It is responsible for creating change over time. As a botanist, key writers and users must understand this variation. Variation lurks at different levels. And because of this, it is just vital that you always measure as many replicates as possible. Do not settle for measuring just one leaf. If you have a whole tree, measure as many leaves as you can and take the average. Or get some range. Measure the heights, different petal numbers. You've already kind of learned this by dissecting flowers. You've seen variation. You've seen mutants. You want to know what the norm is as good as possible. So we're talking all about within a species right now. This whole lecture has just been talking about within a single species. Why does all of this matter? How much variation can a single species hold is another question you might ask. So say we've got these kind of different populations of a species and they overlap in most of their range. So here, by similarity space, I just mean, oh, these species, they kind of overlap in these traits and these traits. Oh, and these populations here, they overlap in those traits. But notice, these two don't actually overlap with one another. These two might be considered subspecies within a species. Here's an example of one that you know. You know ribes, but you don't know the one that looks exactly like this. You're more familiar with this one. This is the one that we have all over campus and the one we've dissected in class, and I think we had it on a quiz as well. I only had you know the genus level, ribes. But these are both species of Ribes sanguineum. They don't really overlap much in their range, and notice the pretty drastic color of the flowers here. In Ribes sanguineum, variety glutinosum, you've got those white flowers mixed in with the lighter pink, while variety sanguineum has much deeper pink flowers throughout. Well, these are different varieties, yet they're in the same species. Here's a more concrete example of why species thing matters. Erysimum menziesii is a very special species of plant that we have here in Humboldt County. The subspecies Eucalyptus is what we have here, and it has always been known to be extremely rare. There's only a few populations. Well, a couple of decades ago, some people realized that a close relative, which they were calling a different species, Erysimum consinum, that's kind of widespread throughout the Pacific Northwest, they actually realized that it's a subspecies within the same species of our Erysimum menziesii. And so although it had been considered a rare species, now that this other whole species was being incorporated into the species, our rare species that was so small originally was now much larger, and it did not fit the rules for the Endangered Species Act. Because it is, of course, the Endangered Species Act, not the Endangered Varieties Act. Okay, so we're going to kind of be talking about species concepts and more issues with species throughout the rest of the semester. Last thing I want to talk about is kind of sources of variation. Where does this variation come from? They would have a genetic difference. So when we're thinking of even, say, a tree or a shrub, that's actually kind of, you can think of it as a population of meristems. Each one of those branches is going to be kind of on its own trajectory, if you will. And you might see differences on a single individual for that reason. Or you might see variation within a population. Again, because sex, which just recombines different genes into the variety of phenotypes that are expressed that we see as variation amongst individuals. We call the environmental component of variation the phenotypic plasticity. So this is when a single genotype, a clone, say, is expressed differently in different environments. This would be the variable expression of a single genotype in differing environments. Remember that word genotype from, say, Bio 105 or something? This is the genomic component of any trait that we have. So not what's expressed, but what you have kind of coded in your DNA. That's what a genotype is. So when that DNA code can be expressed differently in different environments, we call that phenotypic plasticity. Okay. So say you wanted to figure out, well, why do these two individuals vary? Let's just say we've got individuals from different populations, but within a species, maybe it's some buttercup flower. And at a lower elevation, so kind of in this example here, at a lower elevation in this example, our buttercups are able to grow much higher. The mean stem height is much taller than individuals that grow at a higher elevation. Their mean stem height is significantly lower. Okay, so maybe we were wondering, well, why are there differences between these two populations? Is it because of the elevation? Or maybe these two populations just have kind of different genetics, and that's what's causing the difference in heights. Well, to figure out which one it is, we need to do this thing called a common garden experiment. So we need to bring individuals from the high elevation population and individuals from the low elevation population to a middle elevation common garden. And we need to plant them both into the ground and see kind of how do they grow. Well, if you bring these individuals to a middle ground and you bring these individuals to a middle ground as well, then they're kind of both growing out of their normal environment and they're growing in the same environment, hence common garden. Well, if they retain their height, the height that was found at the original population, at the original elevation, then we can conclude that the differences between individuals seen here is all genetic. Okay. Well, say you've got something kind of more like this. When we bring the individuals from low elevation to the middle ground, the individuals from high elevation to a middle ground, they actually end up the exact same height. So shorter than the taller ones and taller than the shorter ones. Well, then we can say the differences between individuals are all based on the environment. Right? Otherwise, the genetic component would have kind of taken over, and you would have seen these individuals kind of more resembling the individuals from the original population, and these ones, same thing, they would have been taller. Okay. What if you see something like this? Well, this would show that there is a combination of genetic and environmental factors at play that's responsible for the differences we see between the individuals in this original population and this original population. This would be genetic and environmental. Okay, that's all I've got for you for today. Tune in to our next lecture.